Hey biology students, these are notes on lipids, so this would be page 6 of your notes on organic compounds. We have previously discussed carbohydrates, and that was our first organic compound type. Lipids is the second. Lipids are very different than carbohydrates, but they do have a couple things in common. And what they really have in common is that they are composed of carbon, hydrogen, and oxygen, but in a much greater ratio than in carbohydrates. And they look very different from carbohydrates, which you will recall look like little rings of sugar. And so lipids do not look anything like those, as you can see on this page here. They store more energy than carbohydrates also, and they are just a lot more complex, so we'll get into it. So you have your page out, and your notes should kind of look like the screen here. You have some blanks to fill in your pictures. They are contained in animal cell membranes. Lipids examples are fats, phospholipids, steroids, waxes, and pigments. And so I want to start off, this focus of this page is about phospholipids. So if we break that word down, we have phospho, which means for the phosphate group, that's on the head of the lipid, and then lipids, because they are lipids. Phospholipids make up animal cell membranes, and their composition is they have a head here that has a phosphate, a head, and then two called fatty acid tails. And so let's talk about the head and two tails of a phospholipid. The heads of a phospholipid are actually called hydrophilic because they are polar. So you can label this polar here. You can say we love water. That's what phileos means love. Hydro means water. So they are water lovers, literally. And so these heads actually are attracted to water. However, the tails are hydrophobic. So we would call them not polar, but nonpolar, right? So the tails hate water, they repel water, they don't mix with water, they're not attracted to water, they are hydrophobic. And so what ends up happening, this is quite a confused molecule because we have a head that's polar and a tail that's nonpolar. So what ends up happening is when they all get together, the tails face each other, the heads face out, and they form a sphere. And so some might call it a micelle. Some might say this looks just like a cell, even though it's kind of empty. So there's water on the inside, and look at all the heads of the phospholipids. They like water. There's water on the outside. Maybe this is blood, which is mostly water, and the heads are okay with that. And the tails both face inside. And this forms what is called the phospholipid bilayer. Bi meaning two, like a bicycle has two wheels. And so there's one layer here and another layer here. So this is a bilayer of phospholipids because it's all of these in two layers. So that is what makes up membranes, cell membranes, and every membrane that's in your body. So it's really, really important to stress the structure of phospholipid and the fact that the head is polar and the tails are nonpolar because it determines its structure being a bilayer and its function being the membrane around that can encircle any piece of any type of cell or organelle or any type of molecule. Now, a lipid structure, we're going to kind of move away from phospholipids. Lipid structures are, in general, are 2 to 3 fatty acid tails long. And so you're going to circle this here and maybe circle this picture because you have this picture. These are 3 fatty acid tails. And they look like chains of hydrocarbon like we've been making because they are. And that's why I had you make them so you would know what this was and not be really be strange to you. But that's not the only part of a lipid. The tails are one part, and just like a phospholipid, the head is the other. So put a rectangle around this head and put a rectangle around this word. because this is a glycerol head. And so these are specifically called triglycerides. This is a picture of a triglyceride because it has 3 fatty acid tails, which makes it a tri, and a glycerol head, which makes it glyceride, so triglyceride. Let's ignore this stuff down here. It's a little bit about glycerol, but we don't really need it. A typical triglyceride, again, has a glycerol head right here and 3 fatty acid tails. Now notice those are straight. So you may have noticed that they could be straight or bent tails. So let's talk about why. Triglycerides can be saturated or unsaturated. Now when I talk about fats and triglycerides, those are the same things. So you might write that somewhere in case this triglyceride word is strange to you. Fats and triglycerides are the same thing. So saturated triglycerides are solid at room temperature. I like to say saturated, solid. These are things like butter and lard, and they are generally animal fats. Okay? They have higher melting points because they're saturated. Unsaturated triglycerides are generally liquids at room temperature, and because they have a lower melting point. And so there is kind of a diagram here of what being saturated means. So there's just one tail shown, and all the hydrogen that could fit around these carbon are shown here, and they're all there. So we call that being saturated with hydrogen. Like, that's what the word saturated means. It refers to all the hydrogen around this that could possibly be. Now, an unsaturated, in comparison, has carbon chains, but it has at least one double bond. Now, the double bond doesn't allow hydrogen to bond here with these carbon, because this carbon has 1, 2, 3, 4 bonds, and so does this one, and so we can't have hydrogen here. It is unsaturated with hydrogen because of this double bond. So we call it an unsaturated triglyceride because it actually bends this way because there's a big hole here. So these tails are bent. So these tails are straight. Saturated tails are straight, unsaturated tails are bent. And you can see in this picture, the bent tail actually really bends the whole structure of the fatty acid tail. Now let's review at this point. We have glycerol heads of triglycerides and fatty acid tails of triglycerides. Which one is hydrophilic? Remember, hydro means water, phileos means love. So the heads love water, and the tails are hydrophobic. Which one is polar? Remember, water lovers, something that is attracted to water, dissolves in water, is polar, just like water. And the tails are nonpolar. So go ahead and label those in your pictures. So we have two different types of triglycerides. Animals produce generally saturated fats or triglycerides, and plants produce unsaturated fats or triglycerides. You see the bent tail now here, and if I asked you on a quiz, why is this tail bent, you would say there's a carbon double bond, right? And it leaves a space for the hydrogen where there are no hydrogen, and it bends that tail. Whereas here it is saturated with hydrogen. Go back to the last slide if you aren't sure why this tail is straight and this tail is bent. But the point is, this is what it looks like on the table. On the table, these straight tails all pack together really nice and tight, and so at room temperature it is a solid. It stays a solid. Whereas these oils, unsaturated triglycerides, their tails are bent and it's hard for them to pack together. So you can't get as many in the same amount of space, and they're just kind of tangled up, and so they will produce an unsaturated triglyceride that's a liquid at room temperature. All the oils, olive oil, canola oil, vegetable oil, corn oil, all those oils are unsaturated. Coconut oil is a little bit of exception. Between it kind of sits right here, where it like almost can be a solid or a liquid because it's barely unsaturated. Now I'm going to skip trans and cis fatty acids, but I'm going to sit on this for a second so that you can read it if you want to and learn about trans and cis fatty acids. This is the second slide of it. I'm not going to spend time with it either, but if you want to know about hydrogenation and what margarine actually is, the fake butter that you use maybe in your kitchen, and you can read a little bit about this. So that I'm not going to go over it in these notes. It's just too much. Same thing with omega fatty acids, a little bit of bonus information. You've heard of omega-3s, maybe being found in fish oil. They are unsaturated triglycerides that actually occur wherever the carbon from the first carbon you count over, and wherever the first double bond is, if it's in like carbon 1, 2, 3, it's called an omega-3 fatty acid. So that's just for free, but there's a lot of practical application with some of this stuff, and I thought you might find it interesting. Some people say, eat your omega-3 fatty acids in your fish oils. Okay, so we've done two different types of lipids so far. Let's review. The first one was phospholipids. It was on the first slide, and it makes up cell membranes and all membranes in the body. The second one was triglycerides. They could be saturated or unsaturated, and we discussed why and what it looks like and all about it. The third one is steroids. Steroids are in a family of lipids, and they have a big head and they have a little tail. Their job is to moderate membrane fluidity. That means that these are phospholipids. So label those phospholipids and label these yellow ones cholesterol in your notes. And they basically keep these tails straight because it restricts the motion of fatty acid tails. So you can write that down and we'll talk about it a little bit more later. I'm going to skip a little bit of the cholesterol talk and go on to the fourth type, and that's pigments. Pigments are the fourth type of lipid, so maybe you should number those. Pigments are all kinds of things like chlorophyll, beta-carotene, like lycopene. It makes tomatoes red. These are all because of pigments. So it has a head and a tail, and it looks like another lipid. It's just kind of a strange looking lipid. So the word I need to teach you right here is amphipathic. So all of these molecules so far in these notes are amphipathic. I'm going to spell it. It's A-M-P-H-I-P-A-T-H-I-C. Amphipathic. To be amphipathic means one molecule that has a polar end and a nonpolar end. In this case, the polar end is the head and the nonpolar end is the tail, in every case with lipids. So pigments, things that give all your vegetables and fruits color, even in your skin, are lipids. And you can tell because of the way they look. Waxes are the last type, and waxes are the fifth type. And they can come from anywhere, really, natural or synthetic. They have higher melting points than triglycerides because their structures are just so stable, and they are also lipids, so they're insoluble in water. They do not dissolve in water. They're hydrophobic. So I don't spend much time on waxes, but they're kind of interesting. So let's review. The 5 types of lipids in order were phospholipids, triglycerides, both saturated and unsaturated. Then we talked about, what did we talk about? Steroids. Cholesterol being the main example, okay? Then pigments. Chlorophyll is my main example of that. And then waxes also. That's 5 examples of lipids.